Okay, welcome to the neurological or nervous system functions and anatomy section. We typically don't go into the detail of the nervous system that truly deserves our attention, but I highly suggest if you ever find any biopsychological courses or neuroanatomy courses or anything like that, the depth of knowledge that we can go into for the central nervous system, so our brains in and of itself, is truly impressive and very engaging and I highly suggest it. I always thoroughly enjoyed those courses. But for this course, all we really ultimately need to understand is some overall system interactions that occur with our nervous system, and then some specifics to do with our sensory system and our motor system. But first, I was just jumping ahead of myself a bit, but what does the nervous system do? Well, it allows us to move around, but it also gives us things like consciousness, awareness, memory, being able to feel things, perceive things. And again, that's another really interesting course, if the university still provides it, is a course on consciousness. Very interesting stuff for sure. Okay, and our typically our nervous system does initiate movement, but one of the other key factors, very important, high key, that is important with the nervous system is that it maintains homeostasis. So what is homeostasis? Well, it's this maintained kind of physiological Goldilocks zone, so to speak. So what do I mean by that? Well, it's just this perfect zone that's not too much or too little of some metric, let's say, that our body can operate in, and that's where it likes to sit. It's come up a few times where I've mentioned, almost every single chapter we've covered so far, I've mentioned keeping balance or shutting something down a bit or turning something up a bit, and that's exactly what it is homeostasis. So it's the ability of an organism or cell to maintain internal equilibrium. So that equilibrium is really important in that it's that buffer zone that's really ideal, by adjusting its physiological processes to keep functions in physiological limits. So that's what that means. So importantly, things that I have said so far is increases in body temperature, has this homeostatic range, acid-base balance. I spoke about that lots in the cardiovascular, despite trying not to use the term homeostasis because we hadn't covered it yet. Hypohydration, so that's too little hydration, changes in blood pressure and altered glucose levels, which is a hormonal homeostatic range. And so how do we do this? How does your body know, okay, I'm a little too high or I'm a little too low, and then self-correct? Because we don't consciously think, well, maybe aside from maybe dehydration or something, we don't think to ourselves, I sense my blood sugar is too low. Sometimes we can, but usually we have to use, if you're diabetic, you have to use some sort of glucose monitoring device because you can't actually tell. And so to keep it right in that perfect range, automatically what happens? Well, we use feedback systems, and largely that's the nervous system at play. So we have two types of feedback loops, a positive and a negative one. A positive feedback loop will intensify whatever cue is making it trigger, and the negative feedback loop will turn down whatever is setting it off. Okay, so an example would be increased acidity increases blood flow as we continue to work out, and we want that increased cardiac output or blood flow if we're exercising because we need that delivery of oxygen, so that's a good thing. In a negative feedback loop, what will happen is things get turned off a little bit. So an example would be body heat triggers sweating. So the idea is the sweating will help cool the body. So we want to turn down in opposition to whatever the cue is. And I find this little schematic kind of helps. So we have some sort of input or a trigger, as I mentioned, that will go to the it says system here, but the organ or the nervous system, in which case it will cause a change in the output, which is the desired tissue or organ, and then feedback. And so the thing is with positive feedback systems is we don't have a lot of them in our bodies, okay? The reason for that is there's no input all of a sudden in this to stop it, okay? So if everything worked on a positive feedback loop system, as soon as there would be certain triggers in our body, it would just increase and increase and increase all these outputs. And again, we get outside of homeostasis. So we need negative feedback systems where when we have an input go into our nervous system, it senses it. Yes, we get the output to slowly turn down whatever that particular system was, okay? So one for kind of stopping things, one for amplifying them. And the neurons in our nervous system do that. So we're going to take a look at our neurons in particular. We're going to explore a little bit how they work. In this section, you get a little bit of background, but as far as their anatomy, but then we dive into a bit more of how they send signals in the next portion. So a neuron will electrically, so there's an electrical impulse. It's an electrically excitable cell that initiates, receives, and transmits info. Now there's three main important components: dendrites, which are kind of the branches. If you think of a classically diagrammed neuron, and we'll take a look at that, the tree part on one end of the neuron closest to the body is where we receive the information from the previous cell. And then axons are the parts where we send off the information. And so ultimately we get this thing called a threshold amount of electrical signal that will reach a component between the cell body and the axon called the axon hillock. And when that signal reaches threshold, we get something called an action potential, which then is the impulse sent by the neuron. We have three different structural types. Multipolar is, again, the typical neuron that we'll probably imagine in our heads without knowing it. Pseudo-unipolar is a kind of unique one where we use it for sensory neurons. It's more of like a one-to-one kind of transmission thing. Somewhat similar with bipolar, where we have one axon, one dendrite. That's also primarily sensory. And of course, here's our multipolar typical neuron. Here's all the dendrites or the branches closest to the cell body. So we get information in on this end. Once the action potential occurs at the axon hillock, we'll get an action potential down the axon to the terminal end of the neuron. And we will take a look at how and what goes on at these synapses down here in the synaptic terminal. We also take a look in a different chapter at myelin. So this myelinated region is really, really important, as is the nodes of Ranvier. Super fun name. Essentially, they help speed up nerve transition. We'll cover that a bit later. But let's take a little peek at those nerve groupings. So again, this is just a horizontal neuron. It looks to be bipolar. We have a unipolar neuron here. This one is a Purkinje fiber, or a neuron. You might remember that from the heart. And again, the structure should tell you their function, right? When we have these kind of uni or bipolar type neurons, we're really having a quite specific signal with not much input, and then a signal that goes on because there's not too many dendrites and not too many synapses at the terminal end. But if we look at something like a Purkinje fiber, lots of input before we get the signal sent to a very specific place. So we want those to be able to fire very readily. Okay. So taking a closer look at the synapse, as I've mentioned, so again, that's at the end of the neuron after the signal has traveled down the axon. So we have our axon terminal here. You see there's lots of mitochondria there and things called, they say secretory granules, but we know them more as synaptic vesicles or vesicles in general. And these will be filled with something called a quanta of neurotransmitters. And so these quanta migrate once we get a signal. They kind of get a push like the wave machine at the wave pool, I guess, where these will get signaled to fuse with the outer membrane. They're just essentially vesicles that can integrate themselves into the outer membrane. And as they do, they dump their quanta of neurotransmitter into the synaptic cleft, which can then act upon the next neuron down the chain, which may or may not cause an action potential, only if threshold is met. And of importance for us is sensory neurons. So they carry messages from our sensory receptors in our bodies up to our central nervous system, being through our spinal cord and to our brain, so it can be integrated. And from there we can choose to have a motor response potentially. So it will carry from our central nervous system, again primarily being our brain, through our spine, and then out to our periphery, our peripheral nervous system, in particular our muscles, to have a motor response. And that's through something called motor neurons. They have a cell body in the spinal cord, but a very, very long axon that reaches out to the muscle fibers themselves. And so they have really short dendrites to receive information, again, primarily closest to the spinal cord. There are some supporting neurons called interneurons, which connect essentially sensory information from one component to motor neurons in the other part of our nervous system. But there's also within those motor systems, which we'll detail later, is neuroglia. And these are truly supporting nerve cell types. Now, the interesting thing about these neuroglia is they form the myelin sheaths, which you'll come to understand helps speed transmission of signals down the neuron. We have, and they not only provide the sheath, which is kind of like this fatty insulation, but it also provides nourishment and structural support to these neurons. Okay. Synapses we discussed, that's at the terminal end, and the process we've already detailed as well. Okay. Receptors, though, we haven't. It's on the other end, so after we release our neurotransmitter, which are these little proteins that or molecules, I guess would be a better descriptor, cross the synaptic cleft, and they can reach the other side of the cells, which have receptors. Now, if you recall, receptors are these 3D protein structures that sit on the outside of cell membranes, and they have a unique receptor for each neurotransmitter or hormone it receives. Now, unique might be a little deceptive in saying that there can be analogs or chemical structures that are similar enough to those receptors that will bind, sometimes more with a higher affinity, or compete against neurotransmitters to get there. And that's how we use pharmaceuticals like different drugs to be able to cause different reactions in our bodies, is we create keys that may do a very similar thing to the original neurotransmitter or hormone for that target cell. Okay. I digress. Where were we? Okay, so for kind of setting off another signal down to the next neuron, what happens is some of these neurotransmitters will bind to the outer receptors on another cell, and some of them will open some channels, and if we get enough channels to open, that may cause a change in the electrovoltaic gradient on the outside of the cell membrane and cause depolarization. Okay, and we'll get to chat about that in a bit more detail in a farther lecture. Okay, hope that was a helpful intro to your nervous system and neurons.